Evolution, Lesson 3.2, Evolution of Populations. So last lesson we talked a lot about populations and natural selection and how all those different things interact. But now we're sort of going to start to do some problems to actually see it play out in real life. But first, let's build a little bit of an understanding of what evolution in population looks like. So first of all, population is a localized group of individuals capable of interbreeding and producing fertile offspring. Population is also the smallest unit of evolution. So now we're going to learn something called Hardy-Weinberg and populations. Let's talk a bit about Hardy-Weinberg first. So he discovered that frequencies of alleles and genotypes in a population's gene pool remain constant from generation to generation. So every single allele and the genotypes in that population, so remember that same localized group, will remain the same time after time after time. But it has to meet certain conditions. So provided that only Mendelian segregation and recombination of alleles are at work. And what Mendelian segregation is if you remember when we did all those Punnett square questions with the big T, little t, we'll come back to that right now. It essentially preserves genetic variation of a population. So there are, like I said, sort of things that have to go a certain way in order for Hardy-Weinberg to occur. So five things have to be met in order for Hardy-Weinberg to happen. Okay, so let's talk about a little bit about those conditions that have to be met in order for Hardy-Weinberg equilibrium to occur. So there's one sort of thing that we have to talk about before we get into solving the actual Hardy-Weinberg problems. So Hardy-Weinberg describes hypothetical population. So his sort of populations that he's describing here are sort of fake, and they wouldn't be necessarily real in real life. In real problems, allele and genotypic frequencies do change over time. However, like he said, certain conditions have to be met. So let's take a look at those 5 conditions. One of them is extremely large population size. So it means that there can't be a small population size because it wouldn't be fair to the experiment, right? If you just looked at 5 members of a population compared to like thousands, it would be really different for the outcome. No gene flow, so genetic additions or subtractions from a population resulting in movement of fertile individuals or gametes. So gene flow is what actually causes the population to gain or lose alleles. And we'll investigate that a little bit more later, but essentially what you need to know is that for no gene flow, sort of the definition for this is that it is an increases the variation in a population and decreases the variation between a population. The third one is no mutations. So what a mutation is more at the DNA level. So if you remember back to our DNA sort of unit, there's changes in the nucleoid sequence of the DNA, which actually causes new genetic alleles and genes to arise. So to change a base in one gene is usually harmless, right? But it can change the phenotype specifically, which can sort of create a very different outcome, right? And then we also talk a little bit about sexual recombination, which is more important than mutation because it makes it possible, because it makes adaptation possible. And remember, sort of, that's the whole point of this, right? Adaptation, evolution. Then we have random mating, which is really basic, which means no sexual selection, which we talked about already, and then no natural selection. So if you remember, we talked about natural selection, it being essentially survival of the fittest. So differential success in reproduction results in certain alleles being passed to the next generation in bigger proportions. So this is sort of what we call genetic drift. So how allele frequencies fluctuate can reduce genetic variation through loss of alleles. So let's just back up a little bit. We're going to talk about genetic drift first, and then we'll get back into the Hardy-Weinberg equilibrium problems. So genetic drift, like I said, it has to do with that sort of fifth condition, no natural selection in the Hardy-Weinberg equilibrium. And there's sort of two different types of genetic drift, the bottleneck effect and the founder effect. So the bottleneck effect is a sudden change in environment that might drastically change population size, resulting in the gene pool no longer reflective of the original population. So if we take a look at this original population bottle over here, we see that there's a relatively good mix of the blue and the red dots. But after this drastic change and this drastic reduction in the population, we see that there's going to be dropping out more of these little red guys there, see? And then the surviving individuals, we see that there's less of those blue guys, not a lot of the blue ones survived. And then eventually the last sort of bottle is no longer reflective of this bottle because it has way more reds because the blues unfortunately didn't survive. Then we can come over here to the founder effect, which is when few individuals become isolated from the larger population. It can affect allele frequencies in a population. So we can see that this was sort of the bigger population, and then these three sort of guys got separated from it, but two of the brown ones came over and only one of the orange, where you can see it's more even over here. So those are just like some things to keep in mind if you're asked to give like a definition of it, or sort of to say what Hardy-Weinberg, one of the things about Hardy-Weinberg. Okay, so back to Hardy-Weinberg problems, how to actually solve them. So the way I sort of like to think about it is there are 5 steps. But first, let's break it down a little bit more. So if you remember all the way back to when we did those Punnett squares, we talked about big T, little t, stuff like that. So in Hardy-Weinberg, we actually say that big T is equal to homozygous dominant, right? Because it's big. And this is in Hardy-Weinberg equal to our value of P squared. Our big T, little t is, remember, since it's 2 different ones, is heterozygous. And this is equal to our Hardy-Weinberg value of 2pq. And then lastly, our little t, or our homozygous recessive, because remember it's 2 little ones, is equal to our Hardy-Weinberg value of q squared. So let's investigate how we actually use these two. But remember that our dominant is going to always be that big T, or in Hardy-Weinberg's case, the P, and the recessive is always going to be the little t, or in Hardy-Weinberg's case, the Q. And those will sort of come in handy when we talk about the allele frequency, and then this one sort of the genotype frequency. So I write these sort of down before we jump into the actual solving of the problem. So remember that this side over here where it says genotype, that's for if the question asks you for the genotypic frequency, and then this side over here you're solving, you would only find the P and the Q if it asks you for the allele frequencies. So let's do an actual question. So in a population of pea plants, 1% are short, which means they are homozygous recessive. So more often than not, like I'm telling you, every single question, pretty much, like if it doesn't, but you're gonna get way better at it and you're gonna be able to solve it, will give you the value of, remember, the homozygous recessive value is always Q squared. So it already tells us the value of Q squared, which is 1%. And if you know how to write 1% as a decimal, it's 0.01. So if you remember our steps, the first thing you always do, always, is to find Q squared. And the question, more often than not, will give it to you. Pretty much every single time, basically. Yeah. what are the frequencies of the recessive allele little t and the dominant allele big T? And then it also asks you for what are the genotypic frequencies of this population. So if you remember, if you wrote that down, I gave you for what you need to solve for the genotypic frequencies and the allele frequencies. So how I would go about this question, following those exact steps that I gave you, was first find Q squared, which we already did, so we can put a check. Now the next thing is finding Q. And if you think back to math class, how do you find the square root of something? You square root it, so we would easily just what's the square root of 0.01, and it is 0.1. So now we have our Q value as well. The next step that the thing tells you to find is P. In order to do that, we use this sort of formula, which is P plus Q equals 1. P plus Q will always equal 1. And now we just plug in our values that we know, right? So we're trying to solve for P, and this is just sort of that system of equations that you learned in math and how do you solve them. So P equals 1, you bring this guy over to the other side, so it becomes minus, and we get P equals 0.9. So now look at that, we also have our P value now. The next thing that the steps tell us to do is to find P squared, which, what do? We just square 0.9. So P squared equals 0.9 squared, which equals, if I'm not mistaken, 0.81. So now we have our P squared value. There's just one more thing that we have to do, which is our 2PQ value, which is usually what you do last because you don't want to mess it up and you won't have all the values yet. So 2PQ is sort of like the most important part of the Hardy-Weinberg problem because it uses everything that we already know, and it will always be 2PQ. So we plug in the numbers that we know. So 2 times our P value, if you go back all the way over here, it was 0.9. And our Q value, which we got at the very beginning, which is 0.1. Once we do 2 times 0.9 times 0.1 in our calculators, we get 0.18. And now we have all of our Hardy-Weinberg values. Now it's just a matter of writing it out. So if you think back to that little, what I wrote on the last slide, and I told you to write it down, our big T or our homozygous dominant, if you look back to what I wrote down for you, I said that that was the P squared value. Our P squared value was 0.81. Our little t or homozygous recessive value, also known as our Q squared value, is 0.01. We just look right here because we found it already. 0.01. And then lastly, our big T, little t value, or our heterozygous value, also equal to this 2PQ value in our Hardy-Weinberg if you look back to that chart. So it's 0.18. And now this is what we sort of label when it asks us, we label this our genotypic frequency. And lastly, the question asked us for what are the frequencies of the recessive T allele and the dominant T allele. This is like pretty much the easiest part of the question. Remember that I said that big T was equal to P and little t was equal to Q. So our Q value was 0.1 and our P value was 0.9. And this is our allele frequency. And essentially the questions don't get any harder than this. They're just repeats of this, and as long as you follow those 5 steps that I gave you and you find all of those values in the order that I gave you, and you just think back to the question, what is it asking me? There might be a few extra steps with the math at first to get your Q squared value, but that's about it. Okay, and this is the last thing that we're going to talk about in this lesson, which is genetic variation. So there's sort of three different things in genetic variation that have to do with selection favors certain genotypes by action on the phenotype of certain organisms. So the first one that we're going to look at is directional, which is the one that says A, which is it favors, so like this chart, this sort of wave, it favors individuals at one end of the graph. The 2 is disruptive selection. It favors individuals at both ends of the graph. So it's sort of, that's why you get those two sort of humps. And then stabilizing favors intermediate variation and acts against extreme phenotypes, which essentially means that it favors the middle. So that's why that hump is sort of in the middle. And there could be sort of questions that your teacher asks you, like, is this an example of directional selection? Is this an example of disruptive selection? And essentially what you have to do is you have to ask yourself, is this favoring this end of the population? Is it favoring this end of the population? Is it favoring the middle? And it's pretty straightforward from there how to figure it out. And you can always have these charts in front of you to help you figure it out. And then of course there's that definition on the bottom for you. And that's it.